and transgenic plants, and now she has also started Genomeritin in wheat particularly, and she is the CCPI of Genomeritin project granted by ICR to NIPB. So with this, I invite ma'am to start her talk.
A very good morning to all of you, and I thank Dr. Anil for such nice words. You know, there is a saying that if these words which we hear, if our parents, unfortunately my parents are no more, but if our parents get to hear such words, good words about us, they will be very happy. Because generally, I think you people are comfortable with Hindi? Anyone who's not comfortable? Okay. You learn Hindi fast. That's what they say in Tamil Nadu also. So anyway, I'll try to stick to English. Somewhere if I slip, you please let me know. So parents generally tell you that you are not good. It's rarely, especially in northern part of India, they generally criticize you. So if they get to know that yes, you are getting some good words from some people, I think they would be very happy. So I thank Dr. Anil for this. Secondly, I would label myself as wild card entry. Okay, I am not there in the schedule, but somehow I wanted to be here. So we had discussion with Dr. Anil and he was kind enough to agree to it. So my area of research, like he rightly said, is transgenic, but this transgenic is from 2009. Whereas I started my career in 1993. So it took a lot of time to come to this transgenic stage. Initially, I was in ICR system, it all depends where you are posted. So I was posted in Lucknow in a Mango Institute, the first biotechnologist in that institute who did not have a biotechnology lab, so did not know what to do. Fortunately for me, one of the senior person from Shimla got transferred to Lucknow because of family reasons. Lucknow was his hometown, so he was slightly up the ladder. So sometimes, you know, at S1 it is very difficult to manage things. So then we started building, we were given 2 garages, we started building tissue culture lab. रेनू उसमें से पेन ड्राइव्स निकालो, एक अंदर की पॉकेट में होगा, पहले देखो सिंगल एक, फिर इसका मतलब वो मेरी एक्सचेंज हो गई है. अंदर वाली 2 पॉकेट्स है ना छोटी, उसमें एक में होगा. So then we started working on micro propagation in mango. It was a horticulture institute. We had to work either in mango, guava or papaya. So I opted for the most difficult one, and that was mango. Then because of family reasons, I got transferred to IRI in 2003, started working. तो अगर होगा भी तो BAU होना चाहिए, वो भी नहीं है. नहीं सबको एक में ही कर लो, सब लोग थोड़ा हाँ आ गया. हाँ. So 2003 I shifted here to NIPB. I started. So then I started working on bacteria. The purpose was isolating genes for chitinase resistance, chitinase from bacterial system. So I can say from 2003 actually I am exploring genes. Then 2009 we started working on transgenic, and the yes, go. So as I say, my talk today includes, first is climate change. First let us go to the topic, genes from tolerant plant systems to develop climate smart wheat. Climate smart wheat is just a catchy word. Synonym is temperature tolerant wheat. High stress, high temperature, heat, whatever you may say. But to make it catchy, it is climate smart. Because high temperature and climate at this time, climate change are almost same. So with this, the talk includes climate change, heat stress, agriculture. Heat stress in wheat. My research, I'll be mainly talking about my research work, and you can pick up the points for your project proposal along those lines. From 2009 onwards, can wheat beat heat? Only biotechnological approaches. Breeding is doing very well. I will never undermine them. Breeders are doing good work in wheat, but there has to be different technologies in your basket. You should be ready with the technology so that the need comes, you can use it. As Dr. Anil said, I have been working on transgenic approach, and I am totally novice with respect to genome editing. I am in fact starting with you, attending lectures with you, because I have just initiated work on genome editing under this ICR funded project. And the reason for genome editing is also, I would say, I would have never liked to leave transgenic, but because of public not accepting it, I am ready with my product, which may become dormant in few years. I have my seeds till T4 stage, but if we don't get, it is difficult to maintain. It is not an easy task to maintain your plant material. The other option is you give it to some agency, some institution which maintains it. But they also want total passport data. So for that, even if your transgenics are at T4 stage, you will first have to analyze them completely once again, and then only they will accept. So I have moved on to genome editing till I retire, hopefully. So my area of research, if I define in three major points: search for gene across genomes. Thank you. Functional validation of the isolated genes in model system and your target crop. In my case, it is wheat. For wheat, robust protocol for genetic transformation. So these three are the major areas which I have been handling. And for genes, the source has been different systems which are tolerant: pearl millet, finger millet, Ziziphus, what we call normally as bear in Hindi, prosopis, which is very popular in Rajasthan. It is known as kejri. Then we have also taken few wheat genotypes which are tolerant. One of them, which has been our sort of main source, is Raj 3765, which is to this date a check for heat tolerance. We also worked on Agilopsis speldoides with the aim to search for, again, heat stress tolerant genes. And the target is heat sensitive genotypes. One of the most popular wheat genotype HT2967. It is very, very popular among farmers, but it is heat sensitive. So we were targeting that genotype, but recently in this specially genome editing project, we were advised by the breeders. One thing more, my selection of genotype, target genotype is totally based on breeders. Sometimes I am asked in some meetings, in some projects, that have you assessed this? Is it heat tolerant? But when a breeder tells me that this variety is released as heat tolerant, I do not doubt that person. I take it as such. Some preliminary experiments we do in our lab just for our confidence, but not to disprove them or prove them. So I totally rely on them, and in many meetings I have taken this, put forward this point, that I am not a breeder. If a breeder tells me that this variety is released and this is released as heat tolerant variety, I will go by that. I will not go for all, going in for testing and all those things, growing them in field, etc. So, but 2967, in recent times it has had, it has become susceptible to wheat rust, especially yellow rust. So for genome editing, we are taking another genotype as per the breeder's advice. The genotype which we are taking, 3406, it has a background of 2967, but it has 2 genes for rust resistance. So we are taking that. So now, why do we want to search for genes? First, this is a known fact, population is increasing, itna million food chahiye hoga, ye bahut sun liya. And it is fact. It is same. I won't be telling you anything new in this regard. We need to double food production by 2050. For that, the land resource is limited. We have to maximize the productivity. For maximizing productivity, there are major bottlenecks, abiotic stress, biotic stress. We have to tackle. Tackling biotic stress is comparatively slightly easier and difficult both. Biotic stress is easier because generally it is single gene governed trait. But it is difficult because there are 2 living organisms involved. Whereas in abiotic stress, it is generally a quantitative trait. The advantage is you are handling only one system. Because in biotic stress, you have to take care of 2 living systems. So these are different stresses which reduce your yield. You are not able to attain the maximum yield, or what we call as potential yield of the system of the crop. Now climate change. We all know climate is changing. When we were kids, in summers, 42 was the maximum. For 2-3 days in Delhi, if 42 degrees centigrade reached, it used to be very hot. Water coolers were very effective. This time we had nearing 50. So you can see, and if I see, this is a gap of 25 to 30 years only, not more than that. So if in 30 years it has increased so much, that means it is actually taking place. It is not a myth. 15-20 years back it was climate change is a myth, ye to scientists ne bana diya hai वगैरह करके. It is no longer. It is a fact now. And there are different causes which have been identified, which have been publicized also. External factors, internal factors. Major consequences are also well known, well defined. But why are we worrying about climate change now? Because as they say, climate change has been taking place. It is because of the rate at which global climate change is taking place. Like I said, in ecological terms, it is really very high. It used to be very slow. It used to take place, but it was slow. The second is, it is human induced. So we can do something about it. That is what anthropologically induced, what they say. Then climate change and Indian agriculture. there is a clearly a negative response of crop yields to increased temperature. Studies have indicated 10 to 40% loss in crop production in India by the end of this century due to global warming. Greater loss expected in Rabi as compared to Kharif. Every 1 degree centigrade rise in temperature reduces wheat production by 4 to 5 million tons, which is a big number. The negative impact of high temperature will certainly, because they say, will outweigh the possible benefits of high carbon dioxide. They say if it is high carbon dioxide, high photosynthesis. But the negative impact of high temperature is going to nullify that. So we will not have much gain from high carbon dioxide. Now we come to heat stress and wheat. Like I say, can wheat beat the heat? We are working for that only. You just look at this. In 1924, it was stated, this is a paper, it is stated in a publication, wheat production in India is a gamble with regard to temperature. 1924. And it stands true even to this date. In 2011, we had sown our seedling stage, I am talking about. In November, we had just a small plot. We had sown the seeds for our experiment purpose. Purpose was to give stress and see what is the status, different genotypes. For 10 days, it was high heat. 1 week or so. All our seedlings died. Now you see, this is a small experiment of a scientist who is getting salary, who is getting funds to carry out that experiment. Now put yourself in the position of a farmer. What that person must be going through. I think it was in 2012 or 13, we were asked by our funding agency there was a hailstorm, lot of hailstorm. So we were asked by our funding agency to visit the farmers. So I went to the farmers in the nearby Gurgaon area, and I told them that I am doing bajra, pearl millet. Bajra का कुछ लेकर उसको गेहूं में डालेंगे, we'll put it in wheat, and then wheat will become tolerant like bajra heat. My husband happens to be an extension person. Evening that farmer called, my husband had said कि मैडम वो जो गेहूं बोल रही हैं बाजरा वाला वो कब तक आ जाएगा, मैं अपना एक प्लॉट दे दूंगा. So when we sometimes, when people say कि farmers are not receptive to technology, you give them good technology, they will take it. They will take it when he is in advance booking, while I am still at the stage of searching for genes. So you can understand their stress, farmers' stress. So with that, we move ahead. Okay, now to 1924, 2010. 1 day of high temperature at flowering stage can cause a 25% reduction in wheat yield. Studies have shown that for every degree rise, this I have already stated, this is 1989. So this is a problem. It is a problem. Many times people say it is not a problem. It is a problem. Abiotic stresses in wheat are a problem. For drought also they say many times, wheat to irrigated hai. Wheat irrigated Punjab mein hai. Wheat irrigated Madhya Pradesh mein nahi hai. So we have to tackle these problems. Heat stress on agriculture would intensify in coming years. These will affect agriculture productivity and food security. Naturally, when you do not have, we are able, as a country, we were able to sustain COVID only because of our agriculture. I think everybody would agree to that. Our farmers were working in the fields. We were not loss of any food crisis was not there. So whenever there is a food crisis, it will not only lead to economic imbalances, it will also lead to social imbalances. All social problems are linked with economics. So we have to be careful that we do not reach a stage where there is food crisis. Like India was called Prime Minister with the begging bowl. Are you people aware of that? Indira Gandhi was called as Prime Minister with the begging bowl. She had gone to ask for food. And I think you people have not, you won't know. We have even seen that wheat came from Russia. It was red in color. We have had that red roti also. So crops for the coming years need to be, I will concentrate again on elevated temperature. They should be able to grow at that temperature. So what are the solutions? There are different solutions. First is in nature. Nature, the species, the plant is a very adaptable system. Species may disperse to more hospitable habitats. Phenotypic and physiological plasticity may allow species to tolerate the new conditions. Third, they may adapt to the new conditions through genetic change. That is the power of evolution. The plant will also do it, but again, the pace at which the stress is coming and the pace at which plant can take care of it is very different. It is not in equilibrium. So that is why we have to intervene. Solution, we need to develop genotypes that are tolerant to high temperature stress without any yield penalty. No effect on yield. One of the approaches, development of plants using genes for high temperature stress tolerant systems from land races, wild relatives, tolerant organisms across species, genera, or even kingdom. When I say kingdom, we have taken genes from microbes. So this is, in other terms, we call it bioprospecting of genes or prospecting of genes for thermal tolerance. What is the gene source? Which gene from where? These are the points which have to be taken care of, which I have already said in this thing. Land races, wild relatives, tolerant organisms across the species. So we look for genes from these systems, and these genes should be tolerant to heat stress. Why do we need to move away from breeding? I would like to make this clear over here. We are not moving away from breeding. Like I said, we should have all the technologies in our basket. Breeding is also successful. It has its own advantages. One slight what is happening in recent times, we are not maintaining our land races or ancestral cultivars very properly. As a result, the gene pool is becoming limited. For gene pool, for genes, that is why we have to go to systems which are not sexually compatible with our crop. In case of like for wheat, we are going to related. Pearl millet is a Poaceae family, same family as wheat, because we had tried with Ziziphus and Prosopis also, and we are doing it, but some meetings, seniors advised us that don't go too much far. Though if you look from that angle, from BT is from a microbe and has worked successfully well in cotton system. So, but this has to be explored. To be safe, it is better to go to a system which is slightly linked with your target system. Now, what is the approach we used to take or we are taking? This is one of the approach I am going to talk about. We look for ESTs, express sequence tags. You can get ESTs by different ways. When we started in 2009, we did not have much fund, so it was cDNA library. Then थोड़ा पैसा आया SSH. फिर जब ज़्यादा पैसा आ गया तो transcriptome analysis. So this is how you move. But that does not mean that you have to do only transcriptome. Yeah, you have to do, you can even find good genes with cDNA. I have found many good genes from cDNA system. Then we analyze these ESTs, do full length. In cDNA there will be a problem, you will get small contigs. Transcriptome, there are chances of getting a full gene. Then if you don't get full gene, go in for race. We have done this, we have got good genes through this system also. Cloning, validation of differential expression under biotic stress by QRT-PCR. This can be done when you are analyzing your ESTs also, so that you choose the EST, the gene, based on your this differential expression. Validation in model system, agrobacterium mediated transformation in wheat. This is the overall gist of the work which we have been doing. These are the genes which we have cloned successfully. They are submitted in NCBI. ESTs also submitted. Then transcriptome data also. Transcriptome data, recently we have done one more, which is also to be submitted. So this is a sort of a genomic resource which we have with us. This can be taken up for any gene from this. In fact, we have also given, I'll come to that, some genes to other organizations. In between cloning and validation, we also clone it in E. coli system, and we try to see whether the gene is functional or not. In fact, we have even tried to give stress to E. coli, and that E. coli, after transferring of this gene, our target gene, was able to grow at 45. So that means that gene is working. These are the genes which have been functionally validated from different systems. Ragi, wheat, this is a tolerant genotype, C306, prosopis. Then again, these are from bacteria. This is from pearl millet. Most of these papers are also published. They are, I think, except for microbes and MIB, others all are published. So this is just overall, I am showing some random cases. BZIP is a gene from ragi, which was isolated from ragi through SSH, and then we have validated in tobacco. This is a work of Dr. Amol, who was a partner in that project, and we have a patent of this gene also. This is granted on 20th, this has been granted 20th December 2023. On the similar lines, these are two genes, CSP-A and CSP-B. So cold shock protein, which is actually a cold shock, but it works very well in heat stress also. This has been transformed in Arabidopsis, and these plants are growing very well. This has been validated and it is under publication. This is work of Dr. Kanika, who had also given a lecture to you. Amol was also here. I think yesterday he was here. Now this is a gene from my lab, P5CS. This has been accepted and it will be out, it is online available, This is from pearl millet. This is a proline biosynthesis gene, P5CS, and validated in tobacco. This is in transgenic research, and it is, this gene is worked very well. Many of these genes we have also taken to wheat system. This is overall, I am trying to give MIP, STI, this is published in PLOS, APX. This we had not published because we wanted to release this as transgenic. So this now, but now since we don't have much hope, so we are publishing this now very soon. CSP-A, PCR analysis of T0 plants and southern hybridization of T0 plants. This is what I was talking about. There are few genes which we have shared with people and they are also working on it, but I really don't know what is the status because they would also have stopped working on transgenics. Now we come to genetic transformation of wheat. Wheat genetic transformation is a big hurdle. Like that day Dr. T.R. Sharma told you that there are three things which are very important for genome editing. Your selection of gene first, validation of that gene in any system, and your transformation protocol. In case of genome editing of wheat, we are very confident because we have really worked hard. We started in 2009, like I said. Transformation wheat we started in 2011 and we were able to publish the paper in 2019. It was really difficult. We started with different systems. You know, actually the problem is when you people say, "Arey usne to kar liya, uski lab mein to ho gaya." When you start working in tissue culture, I will not be ashamed to say that it is mainly person based. If even if it is working in your lab, if that student leaves, next student faces again so many problems. But now for our lab, this transformation protocol which we have standardized, it is working. The next student has been able to do it. So now I am more confident. Otherwise sometimes, you know, then there is another thing people will say, अगर आपका protocol अच्छा है तो बाकी लोग क्यों नहीं लेते हैं? I will not put up a stall and say कि मेरा protocol अच्छा है, please आके लो. Those who have read my paper, those who feel they are confident, they will come. They have taken the protocol from me. It is not, I have not gone in for patenting of the protocol or anything. But yes, some people don't, they want to try themselves. Because maybe people have faced failures taking others' protocol. And in case of wheat, there is Dr. Kulwinder Singh in Washington, USDA. He had come over here in India and I personally went with my student to meet him. He gave me everything. He wrote the whole protocol. He had copies of it. He gave it to me. It was in his handwriting. And once he gave it to me, then he said, Jasdeep, I will tell you one thing. I am guaranteeing with Bob White. So Bob White is a germ genotype of USA. Wheat में सबसे major problem, I think most of the tissue culture crops have this problem. It is genotype specific. It may work in one genotype, it may not work in another genotype. And in case of these systems, mature embryo has the advantage, it is available throughout the year. कभी भी इसका seed ले लो, इसमें से mature embryo निकाल के tissue culture कर लो. Immature embryo, there is a window of only 15 to 20 days. But if you see result wise, generally this gives you better results. Shoot tip infection, which we are doing. Floral dip is also like, floral dip is Arabidopsis is floral dip. In wheat also people have tried, some have got results, we did not get. So we concentrated on shoot tip infection, which worked very well with us. We also tried, like I said, it is genotype specific. You can see from this data. Look at the variability. It is so genotype specific in case of wheat. Then we also tried similar genotypes in different systems, different protocols which we had followed. For us, Implanta worked best, so we were confident and we moved ahead with this. This is our protocol. This is published in 3 Biotech, where we allow the seedlings to grow for 1 or 2 days. Then we inject, we prick, inject the agrobacterium, which has our gene, and then we allow it to stay in that culture for 1 day, ताकि जितना जा सके चला जाए, and then we go in for growing these seeds as such. इसमें थोड़ा सा loss होता है. If you have taken 100 seeds, maybe 90 seeds will actually grow. Some seeds get rotted or something happens. Now we allow them to grow. We don't do any screening at this stage, which our protocol of 2019 says. We harvest these seeds, then we do the screening. Whether it is hygromycin, in our case it was EPS-PS, a herbicide, because that was the gene which we were taking as marker gene. And those which are tolerant, they are our putative transgenics. We allow them to, these are only transformants. We call them transformants. So the screening in our protocol is done at T1 stage, not at T0. So this is all the process of screening. Southern hybrid, again we face lot of problem in southern hybridization. If you, for rice, if you take 3 microgram of DNA for transformation, in case of wheat you have to take 40. You can just imagine, it's a very big genome. So, but now our protocol for this is also standardized. We are successful with it. That is why, you have to go to different things. If you're not getting southern, we tried this dot plot में screen करके उसमें से थोड़ा ले लिया. Then we also tried this expression analysis of EPS. This kit is available. So now when this is expressed here, so then we should get southern. Then we move more confidently. 2023, we published another paper of transformation. The problem which we are having, because science is evolving, it is not, you should not think कि हो गया तो खत्म. Our protocol had one major problem, screening at T1. If you have 100 plants, each plant minimum gives you 20 seeds, 20 to, if one spike. If it has 2 spikes, 3 spikes, you can imagine. So 100 into 20, 2000. So you have to, minimum, you have to screen 2000 plants. And 100, because our transformation efficiency is around 2%. So we need a larger number of transgenics also. It is around 3%, 2.65. So we did what? Oh, sorry. We did screening at T0. We did only PCR. PCR का मतलब ये है your gene has gone. So transformation has worked. Now biology will work. Whether it goes into the seed, it goes into the flower, wherever it goes. So we are doing screening at T1. So we reduced our number of plants to be taken to T1 stage. Screening at T0. And so this paper is published in Molecular Biology Reports. And so now we are using this protocol. This published in 2000 was carried out in 20 COVID period का ये काम है. These are few transgenics which we are ready with, and this is APX from Raj 3765 to HD 2894. Similarly, this is another one from this is same. Now these are few transgenics. I am showing southerns so that it is confirmed they are transgenics. These are the facilities which we are using for growing plants. Phytotron, Phenomics, then our own net houses. Now another approach. This is still transgenic. We are moving now transgenic only, but not taking the gene from the other system so that we get some acceptability. We take a gene from ragi, look for synteny in wheat, and then transfer it to a wheat genotype which is sensitive. Example is, I had talked about BZIP gene from ragi. We looked for homology and we found that it had maximum synteny with BZIP60 of wheat. So now we are cloning this BZIP16. Meanwhile, we have also seen the expression of BZIP60 in different genotypes of wheat. And this Raj, like I said, stress, this is a check for heat stress. This is WR544. This also you can see it is responding well. This gene is overexpressing. C306 is also a drought tolerant, and there are always crosstalks between different herbitic stresses. So this is also good, doing good. So this gene we have taken and we took two stages, flag leaf and seedling, and at this pre-anthesis we call it. Post-anthesis, 2 tissues, flag leaf and spikelet. So this gene has been cloned. Among these 2 genes, Raj 3765, BZIP60 and 2967, it is only 48.6% homology. This has been transformed into wheat and we are in the process of screening. T1 screening has been done. T0, 153 are there which we are taking further to the next stage. Similar is in case of pearl millet gene, ASR3, homology in wheat. This ASR3 is a gene which was isolated from pearl millet. This is again SSH library, different genes were assessed, and we found ASR3, which was quite good performer. This was validated in Arabidopsis. Different biochemical tests were done, and this gene was good performer. So real time was also done, and then now we are looking for synteny with wheat. This gene has also been isolated, and it is showing maximum homology to wheat, ASR 4D. In pearl millet it is 3, ASR 3, in wheat it is 4D. And this transformation is going on. Again over here also we had done real time. Before we move ahead with taking that gene, we do real time to just have an idea, yes, the gene is performing. It is just a slight confirmation, it is not confirmed. So this we are moving ahead. Now genome editing, why? I have already answered that question, why are we going for genome editing? First, it is a new technology. We should be aware of it. We should fully utilize this technology. Second, transgenic public acceptance is a problem. So we should have something where we can do some biotechnological approaches in a short span of time and get good products. I'll finish fast now. This is one approach which we are taking for genome editing. Transcriptome data we have in wheat. We have looked for negative regulators and analyzed for differential expression and seen how, like you see this Raj 3765. Under control conditions it is overexpressed, the gene, but under heat stress it is down regulated. So that means it is a negative regulator. In case of HD 2967, which is a sensitive genotype, you see slightly reverse condition. Similarly, in case of HD 3271, which is a sensitive genotype, again you see slight different condition. So that means they are negative regulator for heat stress tolerance. This is one approach we have taken. The other approach which is there in this project, again synteny based. Look for genes which have been confirmed as negative regulator and are performing well in other systems. Search for the homologue in your system and then do genome editing accordingly. This is development of genome edited crops. This already I think you have been dealt very well with, so I will not go through this. This is one example, GABA enriched tomato, which is a CRISPR edited food which has come to the market, and this GABA is known for producing a calming effect. This is in Japan. You must have seen this paper. Similar lines pe vacuolar iron transporter gene in rice. This is just to prove again that this technology also works. Whenever somebody puts a doubt on the technology, try to defend it. As scientists, your role is to defend. We have to be more vocal. We lost the case of transgenics because we were not vocal. You have to have full faith in your technology and you should defend it. These are slight differences which I have worked out. The major which I find is minimal regulation in case of genome editing. This is my USP for genome editing. रेस्ट तो ठीक है, सब कुछ ठीक है, हो जाता है. ब्रीडर्स भी बहुत सक्सेसफुल हैं, दे आर नॉट लैगिंग बिहाइंड, एंड स्पेशली मार्कर असिस्टेड ब्रीडिंग हैज़ डन वंडर्स. सो these are few genome editing crops which are at different stages, and that's all. Thank you. बोलो. July. Now you are shifting to July.
We are going with the same process. So we have identified some of the lines from the 80 germplasm which we have the 3K panel, and we have gone for a field screening.
Hmm.
So field screening along with some genomic screening as well.
Yeah.
So ma'am, I just want to know how you screen your plants in field to just know about whether they are heat tolerant or heat susceptible.
Like I said, I do only screening of transgenics.
Okay ma'am.
For other germplasm, wherever I am associated in some project, my role has been biotechnology. I have not tried to intervene in that area. They do the screening. Like for example, I had one for Agilopsis. I was with DWR, IIWBR. So they did everything. In between, I used to go and visit just to get a feel of it. Then they identified two lines. Ki, take these two, and they also mostly they just tell me the exact because it was not a released
Yes ma'am.
cultivar, so they just told me the number, your IC numbers,
Yeah.
that way. So I have worked like that. Whether I am wrong or right, I don't know, because I feel it is better to do what you know best
Okay ma'am.
instead of they do the screening. Even for transgenics, when at one stage we had given to them
Okay ma'am.
कि इसको स्क्रीन वो कंट्रोल्ड कंडीशंस में, बट अब तो बहुत ही ज़्यादा
Yes, ma'am.
रिस्ट्रिक्ट हो गया. इट इज़ नॉट पॉसिबल. इट हैज़ टू बी डन इन लैब ऑल्सो. दैट ऑल्सो यू हैव टू हैव IBSC अप्रूवल फॉर डूइंग एवरीथिंग. सो
Yeah. Thank you so much ma'am.
staggered sowing, they call it staggered sowing. They different one week का difference देकर in field.
We go for staggered sowing just because if we need to take some varieties for the crossing program, so it is very difficult to get fresh panicles which are having good pollen content. So that's why we have this particular gap of 7 to 9 days or 10 days,
One.
so that we can easily take those varieties forward.
Staggered. No, but her question is more related to how are they assessing it in the one is staggered. वो तो आपको दिखेगा. एक जो टेस्ट कौन से करने हैं, कैनोपी टेम्परेचर वगैरह वो सब शी इज हाँ हाँ
Because I have gone through some papers,
हाँ
but it is not yet been standardized in how we are going to take up the screening in field. We can do it in lab in controlled conditions, whatever we can do.
But in wheat they do it in field also. I think these are, as far as I remember, ये different tests जो होते हैं, Lycor से जितने भी टेस्ट आ जाते हैं, because these rice people many time I have Lycor in my lab, so they take my Lycor during this season to assess heat stress. हाँ, it has lot of parameters. You can like different chlorophyll content भी on field आ जाता है उसका. Then वो canopy temperature कितना है, all those things can be worked out. Huh? Okay. She wanted to ask.
Hello ma'am. So as you showed that using the RNA-seq data we can select a gene of interest. So but based on just RNA-seq data, or maybe if a gene is expressed in a suppose as you showed that if gene the role of gene is determined in maize, but we want to work in wheat, so we can take the same gene and find the homologue. But that is for in that case we are not 100% sure, even based on the RNA-seq data we are not sure that that gene is playing that role or not.
Agreed. That is why I said that first is differential expression. You will have different targets.
Yes.
If differential expression is giving good results, you will be confident. And then, like Dr. Sharma also said that day, functional validation of the gene is very important. You should be very sure, yes, your gene is working in your system. Then only you should go in for any biotechnological approach.
Okay, so first we need to
Functionally validate.
Okay, thank
If you are comfortable with the target system, it is very good. But if you are not comfortable, you can do it in some model plant.
Ma'am,
Yes.
I just want to know, breeding approach is the main basic for genome editing. Means whatever breeders still now concluded or explored the area of genome in major crops or model crops, it is the main base for genome editing.
No?
Yeah, without information we can go on genome editing.
No, that's what it depends on you. If you are confident, you can do the screening yourself.
Yes.
Because you should know what is the role of that gene. Whether editing it, whether deletion, addition, whatever. We are allowed only with addition, deletion or prime editing, right? 3 only we are allowed. We are not going for SDN3.
Yes.
Yeah? So if you are sure that any change in this gene will lead to the plant having your desirable trait, you can do that.
Okay. Just like ma'am, I am working on ragi ma'am, finger millet. So now I have transcriptomic data like DEGs.
For which stress?
Heat stress ma'am.
Heat stress.
So
Okay.
how can I go ahead for the implementation in major crops like wheat, maize?
Acha, you from ragi you want to go to wheat.
Yes ma'am, for heat tolerance.
Then I would suggest this approach. Baki rest you can discuss with others. Synteny based.
Okay,
From
ma'am.
your transcriptome data, you identify the negative regulators
Okay.
and you go in for synteny based and then validation.
Ma'am, which approach is good, SDN 1 or 2 then?
That you have to decide. Because
Yes.
your type. Because and the kind of gene you have.
Okay.
Suppose some deletion, it is more of a transcriptional factor which is affecting many pathways.
Okay.
So if you do one deletion, it may affect many other pathways.
Yes.
So in those cases, prime editing would be better.
Okay.
That you have to decide.
Yes.
Okay.
Okay. Thank you, ma'am.
Thank you. चलिए, ठीक.
Ma'am, when you are talking about the floral dip method in the wheat spikes, so is it like we are wholly dipping the spikes into the agrobacterium culture, or we spraying on it, because the spikelets are getting closed structure, so it's very hard to insert.
We were doing dipping and
Okay.
then spraying also, but we were not successful much. But there is a paper from our institute only in Journal of Biochemistry and Plant Biochemistry and Biotechnology. You can check that. They
Okay.
have talked about floral dip method from our
Okay.
institute only. If I get the link, I will send it to Dr. Anil. He will forward it to you.
Okay, thank you.
Okay. चलिए. Thank you, sir.
So if there is no other question, I would like to profusely thank Pam for
Thank you.
very nicely summarizing all the work. This shows the magnitude of work she has done over the past few years. Thank you, ma'am.
Thank you so much.
Before that,
Arre.
we would like to felicitate you for the same.
Thank you.